Let's talk about meiosis, why it happens, and what's really going on during it. So here are two human beings, a male and a female. How many chromosomes do humans have? Some of you will say 23 pairs, and you are correct. Some of you will say 46, and you are also correct. Because 23 pairs means you have two sets of 23, which is 46. So this couple wants to have a baby. How many chromosomes do you think this couple wants to see in their baby? Probably 46, right? It's a good bet. So women make eggs. They don't really make them. Well, they're born with them. They're born with all of the eggs they're ever going to have. Men are making sperm on a regular basis. How many chromosomes do you think are in those sex cells right there? Some people will say there are 46 in there, and I'm going to show you what would happen. If there are 46 in the egg cell and 46 in the sperm, then when the sperm fertilized the egg, you would have 92. Well, that's impossible. You can't have 92. We need the kid to have 46. 92 is way too many. That can't happen. So what is the process then of taking that chromosome number and reducing it by half? The process is called meiosis. Meiosis is the process that creates your egg and or sperm, depending on who you are. It takes your parent diploid number and it reduces it by half for your gametes, also known as your sex cells. So there you go. That's much better. Meiosis occurred. This egg cell now has 23. The sperm now has 23. And when the sperm fertilizes the egg, you'll have a baby with 46. And that sounds just about right. Because these gametes have 23, we call them haploid, half the number. And this baby would no longer be a mutant with 92 chromosomes, which is good. We don't want baby mutant with 92 chromosomes. All right. So there are two events that happen during meiosis, and they're really important for increasing the genetic diversity in organisms. So the first one is crossing over. And crossing over occurs when two chromosomes that have similar information on them get together and they exchange information. So dad's gene for eye color might get swapped for mom's, and mom's gene for dimples might get swapped for dad's. And it's just a big genetic recombination swap fest. That's why when you look at children, they have some traits from mom and some from dad. It's also why siblings don't all look the same. So crossing over does this, and also independent assortment helps to make this happen. Independent assortment is when the two alleles that make up a gene independently or randomly make their way into various different sex cells. And all of this genetic, the randomness and the combination make all individuals unique. Okay, so let's take a dihybrid cross and we'll show you exactly what independent assortment looks like. So mom is heterozygous for brown eyes, and she's homozygous recessive for no dimples. Dad is also heterozygous for brown eyes, but he is going to be homozygous dominant for dimples. Heterozygous hetero means different for brown eyes. So we must have one capital letter and one lowercase letter. Mom is homozygous recessive for dimples, Homo meaning same, recessive meaning lowercase letters. So she must have two little d's. Dad is, again, heterozygous for his brown eyes. So he must be big E, little e. And he's homozygous dominant. So he must have two uppercase d's. So these two letters represent a genotype, a gene. You don't see them? Well, you see the brown eyes. That's the phenotype. You don't see your gene, which is the letters, the alleles that make up this gene right here. So this is mom's gene that codes for her eye color. And each gene has two alleles. A big, we use capital and lowercase letters. And each of these alleles are going to randomly assort through this process. So maybe this big E is going to randomly assort with this little d. But maybe this big E is going to go with that little d. Maybe this little e is going to show up with this little d, and maybe the other little e with the other little d. Who knows? It's all random. Big E with big D. Maybe it'll be little e with big D, though. This is what random assortment is all about. And now we're going to create our Punnett square, our 16 space Punnett square, because this is a dihybrid cross. And I always put mom's alleles on top. I always work a Punnett square the exact same way. So these are all mom's assorted alleles, and this is what would end up in her egg cells, randomly. The E's and D's randomly assorted into the egg cell. What should always happen is that it'll always be at least one E and at least one D. Now, I'm going to put Dad's on the side. I always put Dad's on the side. Here are his randomly assorted alleles, and those are the alleles that would be in his sperm. So now we're going to do a Punnett square to show any one of these kids could have a random collection of genes. All right, so the last slide was a little crowded, so we're going to do a new slide. It's the exact same problem. Now, because brown eyes are heterozygous, we know that brown eyes must be the dominant trait, the dominant phenotype. Okay. So here we are. We have mom's various eggs and dad's various sperm. And now we're going to start our cross. So we trickle down and we slide to the side. Now, just want to say, it doesn't matter if you write it big E, little d, big E, big D. Just normally, when it is written, it's written big E, big D, little d. That's normal. But both are right. And that's how you do it. You trickle the top ones down into every box below it, and you slide the ones to the side to every box on the side. And that's just showing that this sperm might randomly fertilize that egg, or maybe it'll randomly fertilize that egg. Who knows? All right. So this is how you fill out the whole dihybrid cross. So anytime you see a capital letter, you'll see the dominant trait. So what's the chance of having a blue-eyed kid who has dimples? Well, if brown is the dominant trait, then I can't see a capital B if I'm going to see blue eyes. I have to see two lowercase b's. right? Because you're looking for a gene that doesn't have a dominant allele. Okay, so we're looking for 2 little e's for that blue-eyed gene and dimples. Well, dimples is the dominant trait. So I need to see, do I need to see 2 big D's or could I see just 1 big D? I only need to see 1. I only need to see 1 capital dominant allele, capital letter or dominant allele in order to see the dominant trait. So I could look for EE capital D, or EE capital D lowercase d. All right, so I go through. I'm just going through and checking square by square. Do I see 2 little e's and at least 1 big D? Well, there's 1. All right, there's another one. There's 1, and there's 1. So that's awesome. So how many altogether did I have? I had 4 out of 16, or 25%. And that's why independent assortment also increases the genetic variation in offspring.